Night at Wednesday night at the lab if you want to come or listen in. Jessica Waugh is going to be speaking on pollutants, parasites, and that'd be a good one. And the weather should be nice, and we're going to have the PBS Wisconsin crew there for that talk. All right, now we're going to start this one.
Should I start?
No, we're counting to 10,000 slowly. Put a little gap in there. Okay. Today it's my pleasure to introduce to you Namita Khsjsnchi. Way to go. She's a PhD student with Khsjsnchi at the Wisconsin Institute for Discovery. I'm going to ask her the 5 questions that I ask everybody. Namita, you can answer any way that you would like.
Okay.
Remember, it doesn't have to be true. It has to be believable. You can also demur if you'd like. Namita, where were you born?
I was born in Jodhpur, India.
Where's that?
Jodhpur, India is North India, so it's all the way across the sea.
And where'd you go to high school?
I went to high school. Let's see if it lets me turn this.
Were you going to tell us all this? Okay.
Yeah, I got it planned. I was gonna do initially a map, but I didn't end up having time. But I did high school in Miami, Florida. So I've been in, well, I was in Florida from 2000 to 2015.
You were there for the election in November of 2000?
But I couldn't vote then. So
Did you?
5 years old. That's a good question.
You're qualified. Okay. So where'd you go? And in Miami, where'd you go to high school?
I went to, that is a trick question because I actually went to 3. I went to something called Terra Environmental Institute, which was a magnet school, and I was doing initially biomedical sciences there. And then I decided they're not giving me enough AP classes, so I went to my home school, which is Miami Coral Park. And then the next year, so that my 3rd and my 4th year, I actually did at Florida. What is it called? FIU. I don't know what it stands for. Florida Institute
for High Quality Memory.
I went to FIU, which is a university there for my last 2 years of high school.
Florida International.
Thank you. Florida International University. I really don't remember. And
nomenclature. Yes, okay.
no major in high school, but then afterwards I went to Amherst College, located in Amherst, Massachusetts, and I got my BA in chemistry, and now I am at UW-Madison for my PhD in biomedical engineering. So it kind of all came in circle because I did biomedical science in high school.
I'm gonna ask, what's it take to get a biomedical engineering degree when you don't have an undergraduate?
Oh, I will admit it was very hard to get into schools, especially with my GPA. So so I went to a liberal arts college, and their classes are very tough, especially because I started with organic chemistry instead of chemistry. So it took a little while to get used to how they teach there.
So you majored in masochism. Way to go.
There you go. That's also true. Just a tad. I think it's just hard getting a PhD now. Yeah, but I'm on my 4th year. I have 1 year to go, and you know what? What doesn't kill you makes you stronger. So here I am.
All right. Would you please join me in welcoming Namita?
So I guess, so my research is on controlling CRISPR-Cas9, but before we begin, what do you guys know about CRISPR? Anybody know anything about CRISPR? Oh, yes. Yes. Anyone else?
Got a guy. Just cut off segments and the cell kind of patch something in that you provided.
Yeah. See, you guys are ahead of the game. You studied. I am glad. So this relates back to what you said. It does come from a bacteria. And so this question, is bacteria good or bad? Both. So what doesn't kill a bacterium makes it stronger. So Cas9, as you mentioned, is an enzyme that bacteria use to fight off viruses. And it works not by targeting just the virus itself, but also the bacterium. So it sends the bacterium into a dormant state and makes it inhospitable for virus to reproduce. And this protects the bacteria from mutating the virus that can slip past the other immune defenses. And so CRISPR was discovered in 1996, but in 2011 and 2012, CRISPR was beginning to be used in different organisms, and variations by these two lovely women, Jennifer Doudna and Emmanuelle Carpentier, who actually also won the Nobel Prize in chemistry in 2020. And then in 2012, Feng Zhang from Boston started diving into using CRISPR-Cas9 in human and mammalian cells. So let's see if this works. This may or may not work. Nope, it does not want to work. All right, well, this was supposed to be a video. It's not doing what it wants. So I will just go over what the video talks about. So the DNA in millions of cells need to be corrected to treat many diseases, and getting the editing tools into different types of cells, such as that of many different organs, will need to be tailored. Delivery techniques need to be tailored and packaged properly. And so these delivery techniques must be specific to enter the right cell and avoid the wrong cells, such as that of other organs or even reproductive cells. And additionally, different types of editing tools may be needed to correct different types of mutations. And so I will be, sorry, am I shaking too much?
No.
Oh, perfect. Thank you. And so basically we want CRISPR-Cas9 to come in and fix our cells. And so there are multiple types of gene therapies. Two of the main groups are ex vivo and in vivo gene therapy. So ex vivo editing we can do outside the body, and in vivo is inside the body. And so for ex vivo, there's actually two subgroups, which are autologous cell therapy and allogeneic. Autologous is you can take the patient's cells out, make the edits you want, possibly to correct mutations that might be causing diseases, and then put them back into the patient. And then allogeneic cell therapies is using a healthy donor and getting those cells. If an edit is needed, make that edit and then put it into the patient. And then for in vivo editing, what is usually done is we can put in our gene editing tool in this thing called a plasmid, which is just circular DNA, and put those into cells and have it produce virus that includes our CRISPR-Cas9, for example, our guide, as you mentioned, and our possibly, if you want to put in cells that way, you can as well. So this is used by injecting into the human, and then the editing can occur in vivo. And so while science is rapidly and aggressively being applied, the evidence on safety, efficacy within patients is expensive and limited, especially when it comes to rare orphan diseases. So I actually worked at BioMarin Pharmaceuticals, which focuses on rare orphan diseases. We weren't focusing on the one that I'm about to talk about, but that is where I basically was like, I want to do a PhD related to diseases and then go back to hopefully BioMarin and see what they're up to. So we're gonna take the inherited retinal diseases, for example. So there's about 1,000 to 2,000 patients in the US that have a mutation in this gene that is called RPE65. And from that population, actually, only 7 to 16% have a disease called, it is called Leber congenital amaurosis 10, and it leads to childhood blindness. So it's an inherited disease. And no treatment is currently available for the disease, and it is the leading cause of blindness in children. So the research in general for LCA started in 2008, but it took about 10 years for FDA approval to treat this disease. And it uses a drug called Luxturna. And even then, it is so expensive. It costs like $425,000 per eye to fix. And then moreover, Luxturna is not a cure. It's just to improve vision and slow down the progression of vision loss. And so I don't know if you guys know, but in the last couple of years, Editas Medicine, they carried out clinical trials to subretinally inject modified virus, like I was talking about, doing this in vivo, and it carried genetic material encoding components of CRISPR and 2 RNA strands to guide the protein to its targeted sequence. And so this specific guide was designed to go to the problematic part of the gene, which is that gene is called CEP290, which encodes for a protein needed by the photoreceptor cells that the eyes use to sense light. It was the first trial in the world to directly inject the CRISPR-Cas9 system into the body rather than doing ex vivo editing. Plus, it seemed to cure the blindness for a couple of years before remission. So that's pretty cool. And so there are many different types of genome editors that have been in clinical trials, and a bunch using the CRISPR-Cas9 system, which is the system that I have been using for my dissertation project. And so here are some active technical projects going on at UW. There's a bunch using transgenic animal models and using cell line stem cell bioengineering. And there's also a lot of work that my professor is doing on delivery vectors. So this is using virus to deliver versus non-viral methods to do delivery. And one reason we might not want to do viral related deliveries is because obviously we are not used to viruses, as you can see with COVID-19. Our body will basically create an immune response to them. And once, if we've done the editing with the virus that our body reacts to, we won't be able to do editing again. So I guess I can pause real quick if we have any questions so far.
You mentioned the 2 years for the eye.
Yeah.
Can you speak to whether that, would you get another dose?
I believe they are still confirming if there should be another dose. Yeah. Yes. Yeah. So I believe it's yep, right here. Yeah. And there's been like a lot of work done on that, and they're continuing to do clinical trials on those. It's just that it needs to be, clinical trials are not just short term, they need to be very long term, up to 10 years, up to even like the patient's actual life. Yeah, but they are working on that, thankfully. All right.
You those in Europe.
Yeah, no, there's a bunch in Europe. This is also from 2009 versus 2019. If I'm not actually mistaken, this paper is actually may or may not have been written by someone from Europe. I'd have to recheck. So that could be why, you know, it's biased towards this way. But I think Europe is doing a lot of work. All right. And these are some disease targets that our lab has worked with. Pompe disease, retinal disorders. One big one that we're currently working on is cancer and T-cell immunotherapy and Alzheimer's disease. I actually do not know what idiopathic means. Anyone know what idiopathic means? This was a copy and paste from one of my professor's slides, I will admit that. All right, so it's not related to initially being inherited. All right, see, you guys are teaching me. All right. So why would we want to genetically engineer a cell? So there's many different reasons. I laid out some for humans, therapeutically relevant for diseases, in plants to improve the resilience, nutritional value, and growth rate of crops. And then in animals, it has been used to develop worms that glow in the dark to allow scientists to learn more about diseases such as Alzheimer's. I think that's pretty cool. It can also be used to fluorescently tag a protein to track its expression or intercellular location. And so with that, I'm actually going to jump into one of the ways we've used fluorescence as well as CRISPR-Cas9 to edit some induced pluripotent stem cells, which are also known as iPSCs. So induced pluripotent stem cells are derived from patient somatic cells. So somatic cells are just any cells that are not your reproductive cells. And iPSCs have unlimited growth potential and can be edited via various genome editing techniques, which can then be used for various applications such as the screening of drugs, modeling genetic diseases, or for cell therapy purposes. And I mentioned before that the cells can be derived for autologous therapy or from donor cells for allogeneic cell therapies. And so one popular method that is used to edit iPSCs, as I mentioned, our favorite. Do you guys remember? What's our favorite gene editing tool? CRISPR, yeah, CRISPR-Cas9. And as mentioned before, it has two parts. One is the Cas9 enzyme itself, and then the guide RNA. And so the guide RNA targets the DNA sequence of interest and basically makes a cut. And the resultant double-stranded break can then be repaired by one of many DNA repair pathways called non-homologous end joining, NHEJ, or if you insert a donor DNA. So if you want to correct like a mutation, you can insert like a particular DNA that you want to go into your genome. It would be resolved by a repair pathway called homology-directed repair. Yes. Right. So that is one of the things that actually happened with the Leber blindness that I was talking about. So what occurred was, yes, they made the edit to the cells, but then a bunch of the cells that did not have the edit started increasing. So the hope is that you're multiplying the edited cells, but you're not going to always get 100% of that. Yeah. All right. So, however, this leads perfectly. However, you're going to get a bunch of different type of iPSCs. Some are going to be unedited, some are going to be edited, and then some are going to be edited in places you don't want them edited at all. So this is called off-target editing in the spots that you don't want the edit to occur. And it's important to select gene-edited iPSCs for downstream applications. Oh, is this all just being like, oh, so much information for you? But feel free to ask me any questions. So here came one of the questions that I asked for the 2 years that were initially part of my PhD. Is there a way of adapting a nuclear imaging system to this problem? And we started exploring the literature to understand the relationship between the nucleus of the cell and gene editing. Are we good so far? Questions? Yes. We're about to go through it. Perfect question. All right, let's see. All right. Yeah, you're a mind reader. So to address this challenge, we wanted to develop novel methods to generate gene-edited iPSCs in a rapid and efficient manner. And so recent studies at that point indicated that chromatin structure can have an impact on the CRISPR-Cas9 gene editing. And several groups have used this information to improve gene editing efficiency. For example, heterochromatin, and the presence of nucleosomes have been shown to negatively impact gene editing efficiency, while active transcription in open chromatin and high DNase hypersensitivity increases gene editing efficiency. So these are these two right there. And with this background information, we focused on developing a method to reduce heterochromatin. I know these are words that are just popping out, but I will try to repeat what they mean, what open is versus closed. So to improve gene editing efficiency, we hypothesized that the opening of chromatin should increase gene editing efficiency, and that nuclear imaging will help in identifying chromatin state that is favorable for gene editing. And so we also applied a small molecule called TSA, which is a histone deacetylase inhibitor that causes chromatin decondensation. So we did the overall gene editing strategy that we established, where we first seeded these cells that had this GFP in it. So this green fluorescent protein that we could use to fluorescently label our cells. And we put those in 96 well plates and then gave TSA, so this chromatin decondensation inhibitor, and gave it at various concentrations to promote global chromatin decondensation, which means open the chromatin up so that CRISPR-Cas9 could come into our DNA and start working on it. We also then imaged the nuclei, and this is what we got. So I know this is a lot to take in, but basically the main goal of this is that we can see that our nuclei are labeled with different intensities. And we use basically the heterochromatin intensities, the open intensities over total nuclear intensity. This is probably a lot of information, so I'm gonna quickly skip through it. Yes. Yes. Correct. Yep, so it identifies the nuclei. All right, so let's get back to talking more about how we made CRISPR work in this. So we basically went to hit this green fluorescent protein, and wherever CRISPR edited it, it would stop the fluorescence because we made a cut right there. And so we use that as basically a way to see our editing so that we don't have to do something called sequencing every single time. So sequencing is something that I'm currently doing in my project, and it is very expensive. This is the easier rough and cut method to basically see whether or not you've edited yourself. All right. And we also did something called karyotyping, which is just to make sure that our iPSCs have all of the chromosomes intact. And the chromosomes are happy. We haven't changed anything that could cause like our chromosomes to be mutated in any way. All right. So these are images of the iPSC nuclei after various TSA treatment. So as you increase how much TSA you have, you start to see that there's less green fluorescent protein because Cas9 has come in, cut that green fluorescent protein. And the more TSA that you have, the more open your DNA should be. All right. So this is just in graph form. And we will skip that one. So, whoops, perfect. So we also looked at the viability of the iPSAs with this TSA and noted that cell viability does not significantly decrease until we put TSA more than 6.25. So at 12.5, yes, it might look good, like we've done a lot of knockout, but the cell viability goes down. So more cells are dying. So if we think about doing this clinically, we would have to figure out a good amount of TSA to put into human.
So you determine the viability of each cell?
So we determined it by using flow cytometry. And so what that does is we put a viability dye, and anything that is dead will take in that blue dye. And we would be able to see it. So we don't have to like hand count basically, because this process called flow cytometry will take our cells and tell us which ones are alive and dead.
Let the cells sort.
Yes, you can do self-sorting on that as well. Yeah, you know everything. That is a-okay. I actually had no prior cell work when I started my PhD. So this was very interesting to learn for the first time. And so, yes, so we used flow cytometry to get this graph, and it showed there was no change in transfection efficiency. So that's for our Cas9 to go into our cell. There was no difference no matter how much TSA we added, which is good, because then we know, yes, TSA is not altering the cell from taking anything that we give it. And then guess what we saw when we went ahead and assayed for gene editing efficiency? Yeah, we saw that 6.25 is great at basically editing efficiency. And how we calculated this was like how many green fluorescent protein got knocked out. All right, good so far. I'll be asking that more often because this is like the nitty gritty. The next question we asked ourselves was, since we are globally decondensating the chromatin using the small molecule, so we don't know if it only affected this chromatin, right? It should be affecting all the different chromatin that we have within our genome. We went ahead and started wanting to look at something called off-target sites that I mentioned before. And to test for this, we collaborated with another lab. So this is Dr. Shengdar Sai's lab, who is at St. Jude's. And he determined in his lab that there are 12 off-target sites for our guide. And so we performed sequencing on those sites. Oops. So these are all the off-target sites we had. We had 12 off-target sites. So we can straight up right now say that our guide isn't the perfect guide, but this was not the main goal of the actual experiment to look at like the off-target sites, but we do need to characterize them. Yes.
It is high, bad, and low good, right?
This one, let me check this. Nope. So this one, I know it says OT1, but that's actually the on-target site.
Okay,
Yeah, the rest of them are the off-target sites.
so high and off target is bad.
Yes, low is good. But then you have like OT2, which is kind of high there too. So that's why we were like, let's go through all of them. We need to check for the off-target sites. Whoops. So there was no change in off-target editing efficiency, which was good. So TSA did not make the chromatin that might be part of the off-target like open, or even if it did help it open, it wasn't like enough for Cas9 to come in, do its edit there on the off-target site. And so this is just normalized stuff. And then the karyotype, as I mentioned before, no genomic abnormality with TSA. So we are good.
So,
Yes.
so when you do CRISPR, and you know the sequence of DNA, you then can predict
Mhm.
how many off-target sites are going to be, because you know the total sequence.
Yes, because what usually the reason you would get off target sites, it's you take your guide that you might have, which is for your target DNA. And so you will basically start seeing in your whole genome, where is it that I only have one base pair that's different? And those would be like your off target sites usually. Yeah, because the guide thinks, oh, this is the spot for me to sit, even if it's 1, 2, 3, it could be up to like 5 to 7 different base pairs.
Just for the folks watching, if you move that
Oh.
cursor so they realize that the purple is there.
Oh, yes. That's
What's on chromosome 1?
I mean, good catch. Oh, man.
Very easily.
All right. So we also tested a whole bunch of different guides. So this is the green fluorescent protein guide. Then we tested this VEGF-A guide that's related to angiogenesis in the human body. We tested something called the AAVS1 guide. If you guys didn't know, you have something called the AAVS1 in your genome. We actually got it from a virus at some point. Like this initially was not part of our genome, but it has evolutionary stayed in our genome. All right. And so basically all this tells is that at other loci, gene editing efficiencies increased at 6.25 nanograms per ml TSA. So that is like our happy TSA. All right. So to summarize this part, upon chromatin decondensation with TSA treatment, so open chromatin, chromatin condensation percentage, which is closed chromatin, decreases. Local chromatin accessibility, once again, chromatin opening up, increases. And we have editing up to 3.5-fold more when we add this small molecule called TSA. So that means when I do my project now, if I had TSA, I will get high editing levels. A lot of scientists will do that. They will put in things to get high editing levels. Because remember, if we're doing this for autologous therapy, we want it to happen as quick as possible. We want that edit to happen so we can put the cells back into the patient. All right. So now we'll be going into some of my current research. Any questions so far? So this is well, actually, for gene editing, you would want open chromatin, just so your Cas9 can come in and sit there and your guide can actually find where it needs to go. Yeah, good question. All right, so as mentioned before, while CRISPR has the biggest advantage to target and cut out specific genes, one of the major challenges that comes with it is the cleavage of non-targeted sites, which is called off-target editing. And this could lead to lethal genetic mutations like oncogenesis. So that's tumors forming. So this is just data that was taken by a lab who targeted this VEGF-A gene that I was talking about, leads to angiogenesis. The reason that they use VEGF-A gene in like a lot of the studies is because we have guides that are very specific to edit the site, and then we have guides that we might purposely use to get off-target sites. And I will explain a little bit more in the next slides why I would want a guide that has off-target sites. So here, while the guide RNA does help induce these indels, so these are insertions and deletions made by Cas9, it also induces indels at places that we might not want them. So this is what we were referring to before, that they could have some mismatches. So here's a 1 base pair mismatch. It's just an A instead of a G. And we'll also get the cut to occur there when we don't want it. And this is just an example of, this is the off target right here. If we have Cas9 always expressed in our genome, and we also have the guide that's in there. So we're looking at the P53 guide. Do any of you guys know what the P53 is? Correct. It's a tumor suppressor gene. Yeah. So your P53 gene, you need it to be active because it's suppressing all tumors. But if you put in a guide that will cut P53 and you already have Cas9 in your system, you will start getting more off targets, which is not something you would want, especially in a tumor suppressor gene. All right, whoops. And so, like I mentioned, you're going to get oncogenesis. And so the major challenge right now of gene editing, especially if we want to bring it to the clinic, is that prolonged Cas9 exposure increases cleavage at off-target sites. And the solution is, can we figure out a way to control the activity of Cas9? And so, whoops, what I'm using is a small molecule that basically can control this degron, this degradation little tag on my Cas9. And what happens when I add this small molecule, we can call it drug SM, is it will basically find this degron tag that I have on the Cas9 and start ubiquitinating my Cas9 and then degrade it. Why do you guys think that might be good? Correct. Yeah. So the whole idea is I can now control how long I want my editing to occur with this system.
So how can you remind people what?
Yes. So ubiquitin is this molecule basically that will tell the proteasome to chew up a protein. Anything that gets ubiquitinated, the proteasome will come and chew it up into little pieces.
Like a little label.
Yes, correct. And the thing that makes my small molecule so wonderful is that it can penetrate through the central nervous system and also pass something called the blood-brain barrier. So for brain-related therapies, this would be big. So you can just, do your Cas9 editing, take this pill, Cas9 should technically be degraded, especially if you're using something called the ribonucleoprotein, which is the Cas9 in its protein form, instead of just having constitutively active Cas9.
And how did you find your little green molecule?
Oh, this little green molecule? Yeah, so the degron is actually published, but it's not in a CRISPR-Cas9 related paper. And then the small molecule, they also mentioned that this degron and the small molecule go together. So a lot of times in science, you're just like pulling from other papers and taking those ideas so you can use it next. Yeah. So only one person has to find it. So I'll share, no, I shouldn't share what cancerous disease it is, because then you'll know which one it is. So basically what I did was I made lentivirus. So lentivirus is self-inactivating. I don't have to worry about it. If it does get on me, it's not going to do anything. And it had my wild type Cas9. So this is without the degron. And then I also put in another version, I put it with the degron. And then I killed the cells that didn't have the degron with a selection marker. I'm not going to go into too much detail unless someone really wants to know about that. All right. And I found out that Cas9 protein expression can decrease 45% within 16 hours of me putting this drug in with the degron on my Cas9, which is good because I'm actually going to skip some slides. That is not how good is 45%? Yeah, no, I agree with you. You need a lot more. And let me see if I can actually, now I'm trying to figure out where my marker is. Okay, it's not going to let me do it. I'm going to skip through some slides real quick.
Was that a,
Oh,
go back to your, what looks like a Western blot?
oh yes, it is a Western blot.
Is that a dye or is that old fashioned P32?
P32.
So it's a dye?
As to how I'm getting this,
Yeah.
it's chemiluminescence. Yeah. I know you're saying wow, but I'm going to skip through all of this real quick because I have to show you some. Did I not put it in? I did. There it is.
Well,
This is-
like 1985.
This is what you should wow to. This Western blot tells you a lot. So I put in my drug at hour 0, and you can beautifully see how much it decreases. Once again, you're right, it doesn't go to 0%, but this is only me giving the drug once. And after 96 hours, like Cas9 has continued to decrease. So I haven't done repeated dosage. And then when I take out the drug, I get an increase in Cas9 like that.
How do you take the drug out?
I basically, well, this is all in a dish, right? So I take out the media, I wash it with the liquid, wash it one more time, and then feed it regular media. Yeah, which is kind of cool. But yeah, no, it comes back. So when we do want to make these edits, we can. All right. I'm going to skip through to the fun part because we're reaching like 40 minutes. Oh, yes. No, that's okay. On this one, I actually did not quantify it in percentages.
In the same dish.
Oh, okay. So let's put it, I think I know what you're going at, like how am I doing this experiment? Is that right? So here's this part. So these studies that I initially did, I did not have the guide in there. So it's just Cas9 expression. It's the levels of the Cas9 protein. So I'm not worrying about the editing quite yet. But here I did do editing. So this is to make sure that my degron is not hindering any editing whatsoever, like the degron itself that's hanging on my Cas9. So obviously, if I didn't do something called lipofection to put into my guide, so lipofection uses a delivery system that has lipids that can easily access the cell. So without the drug and without this thing called lipofection or the guide, no editing occurs, which is good. Our Cas9 is not doing any editing. When we lipofect with the drug, we do get editing. And this is good news because I haven't added the drug yet. And it should not have like significant change in editing because that means my degron is not working properly because it's not doing enough editing. And when we put in the drug, you see that there is a 6-fold decrease. We're going from 40% to 7% editing. So this is only for the on target. My guide was too perfect. Remember, I was saying sometimes we would want a guide that does not have, that's not perfect. So here my guide is so perfect that I can't even see the off targets. And so now my next steps are, I have all the data right now today where I have all the off targets. And basically I'm going to sequence those and see, do my off targets decrease when I add this drug? So they're ready for me. And then another next step that I'm going to do is actually edit something called the amyloid precursor protein gene. And this is APP. This has implications in Alzheimer's disease. So this is like big. And as I was mentioning before, I would want to use a dirty guide because the whole part of my project is to see, does the degron decrease off-target editing when we put this drug on? All right, so I'm gonna skip this slide. All right, and then I'm gonna go to CAR-T. Okay, actually, we'll go through the slide. So my technology is great because this degron, which I did not create, someone else created, but we can tag it onto so many different cells, so many different types of proteins, and it will degrade the proteins that we want to. And so I think you guys were excited to hear more about CAR-T therapy. So another project I have in the works is that we attach this dagger to our chimeric antigen receptors, our CAR, so we can tell it when to go on, when to go off. So the chimeric antigen receptor T cells are a genetically engineered form of immunotherapy being used to treat cancer patients. And a CAR is a transmembrane protein that consists of a extracellular expressed single chain variable fragment of a monoclonal antibody. And then it's linked to in the cell to a bunch of other domains. And by using CARs, we can redirect T cells to kill specific cell populations based on their surface antigens. I am not a CAR T therapy person, so I'm not working with CAR T therapy, but 7 out of 10 people in my lab are working with CAR T therapy. And so the overview is that we get blood from donors, and we go through a process of steps so that we can basically isolate the T cells from the blood, and then we can engineer the T cells as we want to include this thing called the CAR, the chimeric antigen receptor. And the idea is clinically, we can put it back into the patient, and if they do have a solid tumor, it would kill off that tumor. And so CAR T cells have seen a great success in treating hematological malignancies, as is shown in this top graph. So for complete response, so this green is big, which is good. There was a response to the CAR T therapy. The y-axis is the number of people treated, and the green indicates a complete or partial response. Oh, I see you being like, oh no, what just happened? I know the picture is a little small.
3 digits on that underneath A.
Underneath A, right there? Yeah.
On the left hand, see how it says 10, 10, 10, 10?
Where? Oh, like here?
Yeah.
Oh, I believe this was a log scale that I might have not put properly. Good catch. Yeah, no, it looks like some of it got covered up there. So these successes have led the FDA to approve 2 different CAR T cell products. I don't know how long it took them to approve it, as we saw, like for the retinal inherited disease, it took 10 years to get FDA approval, though I think that time is getting slowly shorter and shorter, which is good, but then you question, is it actually good because we're not getting enough clinical data? All right. So in our lab, there's 2 main things going on related to CAR T therapy. One is CAR T therapy for humans. So this is to basically get rid of solid tumors. And then the other, which is a new project, is one of my lab mates is actually working on CAR T therapy for canine patients. So she's working with dogs, which is kind of cool. I had never heard of that before, but I mean, it makes sense. Everything can get cancer, that's a lot. All right, we can go into ethics if we'd like to, or I'll leave it open for questions because we're at 48 minutes.
You're good.
All right. So this picture I put up before when I was explaining ex vivo therapy and in vivo therapy, but there's also in vitro germline editing medicine, which is similar to IVF. You take out a cell, do the editing. In this case, we're putting 2 cells together, put it back in. And so researchers from Sun Yat-sen University in China some years ago reported that they had edited the genome of human embryos, and it didn't work, highlighting that the technology is not ready for clinical use. However, the real story was they had edited the genome of human zygotes, a line many bioethicists said should never be crossed. And this ignited a flurry of articles ranging from pop culture to mainstream media to even scientific journals. So here is some, I guess, quotes from people The concept of altering the human germline in embryos for clinical purpose has been viewed almost universally as a line that should not be crossed. I'm fully supportive of research being carried out on early human embryos in vitro, especially on embryos that would otherwise be discarded. So there's been yes, no, maybe. Everyone has had opinions. So, they all met at a conference to have these conversations about the moral fabric of a society, not in merely in terms of we ask questions like, is it a person? But how can we make a deliberate answer, and who do we know how to trust best when it comes to issues like this? And it was a complex, scientifically uncertain area, prediction about the future that may or may not come to pass. And once again, who is responsible for asking and answering these questions that may or may not have consequences? Is it scientists? Is it governments? Is it individuals? And if so, which individuals? And so there are fuzzy lines in gene editing and questions about how it works and where it should be done. And once again, the main question becomes who is responsible for defining these lines? Whoops. And so researchers also looked at laws in different countries about CRISPR embryos, and this is what they got. In the US, does not allow the use of federal funds to modify human embryos, but there are no outright genome editing bars. Clinical development may require approval, so that means at least CRISPR can be used for non-embryo work. The United Kingdom may permit human genome editing for research, but the practice is banned in the clinic. So research is okay, but never take it to the clinic. And then Germany has strict laws on the use of embryos in assisted reproduction. It also limits research on human embryos, and violations could result in criminal charges. So this would be related to IVF, basically at that time, is not allowed in Germany. So while there is controversy about gene editing embryos, we are making our way to at least edit somatic cells and aid in curing pre-existing diseases. And you know what? That's what matters right now. There's so many diseases we could be concentrating on. And I think it's great that we're starting somewhere. And with that, I want to thank my lab members and thank you guys for also being here. I'll take questions.
Is Degron itself a protein or what actually is it?
It is a chain of amino acids.
Okay,
Yeah, bunch of base pairs.
okay. And when I hear
Should
about
be amino acid then.
Well, base pairs make amino acids.
okay, I just got to think how it sticks onto the Cas9. Is Cas9 really kind of a uniform thing, or there's like maybe slight variations in the folding or whatever? And say, well, it's still Cas9, or is
Yeah, so we had a mutant degron, so this was a degron that didn't work. Also, you're right, base pairs is related to DNA and not protein. Sorry, I just realized that.
Oh,
Because you seemed questioning and I was like, you're right.
you just been
Yeah. So we had a mutant degron that wasn't working, that's not supposed to work properly, but it also cut up our Cas9. So we had a truncated Cas9 in that case. But the current Cas9 that I'm testing with the degron, it's not flipped, it's not, messed up in any way.
did people kind of look at making slight changes to Cas9?
Oh.
Or maybe Cas
Oh.
83 or
Oh my gosh, no, a lot. So it would still be called like Cas9, but for example, they have dead Cas9. So that is, if it does rest on your DNA, usually it would stop transcription factors from coming up. So it would be like a repressor. There's a lot of different Cas9s, not even Cas9s. There's like Cas12a. Yeah, there's a bunch. And then they also have like high fidelity Cas9s that they're like, this one works better because it has this one mutation on it. There's a lot
you
of
all
different
remember
ones.
We're old.
Did you say this?
Yes. Yeah, if you're trying to repress like a gene from going on to the protein stage. There's a lot of systems.
Yeah, I know we could get, off-site, off-target cutting. If it cuts in the wrong way, the life could be a disaster.
Mhm.
Just at random, if something does cut in an off-target location, what's the chances that it's really gonna be bad?
What are the chances it's a cut in P53 like we were talking about? There's
I know if it is the one place where you don't want it.
Correct. So that's why usually when they're going through like autologous cell therapy and editing something that's in vitro initially and putting back into the patient, they will check for on and off targets before putting it back in. And I guess one good thing is that they're trying to develop guides that are very, very specific to where you want to edit. Will there still be off-target editing? I mean, some of our sequencing runs cannot tell us that because it could be like 0.0001%, right? So we don't even have the tools to really go down and check to that type of resolution.
Seems like it's kind of a little worrying topic.
Yeah.
You kind of ignore it.
Yeah. Oh, with the edit. Well, the ones they would put back into the patient, they would check for that edit. Yeah. Which I think is the difference. But if we were doing it in vivo, that's where it would get a bit complicated.
But we still have
Correct. And you could technically have a cell isn't going to the genome in a cell is not going to have the perfect edits you want. It may still have some of those off target ones. There's no way that it's, in my opinion, right now, going to be 0%. Like I said, it could be 0.00001%. We're just not going to be able to get that resolution with the techniques we currently have.
So putting the patient from single cell, you've been allowed to divide many, many times. That mixture is what goes.
Yep, that is the one you would want to put in
So
versus
you're always cloning.
one. And that's what takes so much time is that scaling up, especially when it relates to T cells. It's going to take a while. And if you're right, if we do want it to overtake what cells we might already have, we're going to have to do repeated CAR T therapy, for example. So have that repeated right edit in. Oh, yeah.
I can see where having like the allogeneic process would work once something is established. It's working really well because it's such an expensive undertaking to get this stuff all ramped up to the size that you can put into somebody. And so I would imagine you'd have like a bank of allogeneic things for certain needs. But how big would that bank have to be?
Ooh, well, you have to think about, oh my gosh. Okay, so first, I guess I didn't mention too much, but for allogeneic therapies, you would be taking it from a healthy donor. But there is a high chance that your body will reject them if you're the patient. And that's called, like, I believe it's called host graft disease. And your body's basically going to reject those cells, but the bank needs to be, I can only fathom how big your bank needs to be, especially if you're doing repeated CAR T therapy. You would need to be supplying cells like every day, every week. And there's not only going to be one person that's going to come to you. It's going to be like tens of millions. Yeah, I think that is a big question our lab is asking because we're trying to scale up. Right now, we have a program that works to make these edited CAR T cells in like a small machine. And now we're trying to go to a big machine. And it's not like we can size the machine up because all the reagents also are a big factor.
my understanding.
Mm-hmm. Yeah. So I'm not a CAR T person. The main one I know is of our body like rejecting the cells that we're giving, especially if it's allogeneic. Yeah. Oh, yes.
It's her, it's being, the, yeah. So.
Yeah. Whoa. And especially if you're not able to get rid of those dead cells, they're just taking, because they still take up nutrients.
So what's next for you?
What's next for me? That is a big loaded question. I'm thinking about doing a postdoc. I want to be an editor. So I just need to get, more peer reviews in. Don't ask me where I'm doing the postdoc because I don't know. And if all fails, I'll just go work at a biotech company.
And how many talks to the public?
Do conferences count?
No. How many times have you talked to people?
Also, this is the first time I'm doing a 15-minute.
Where did you get your
What?
international unit of power? What? Where did you get your talent? Did you TA a lot, or how did? I mean, you're an outstanding presenter, and this is the first time you've ever done.
No, I don't. I've only TA'd once, and it was not a good show.
Talked earlier.
You know what? I don't know. Probably.
Well, anyway, this is.
But-
What's you done?
But yeah.
I mean, this is a high, I can't believe you get to talk this well, answering all these. questions.
Did you tell them the
Anything you wanna say?
secret
I
of
don't-
so I didn't realize this was a 50-minute talk. I had made my talk for 20 minutes and I had 1 hour to make it 15 minutes. I did skip through a bunch of slides, but based it on time.
Very impressed, thank you very much, appreciate it.
Thank you.
All right,
I get so depressed watching COVID today. It's so much better.
I will tell you the truth. I haven't watched TV in a long. I have not watched the news in a very long time.